Asthma affects more than 1 in 10 children. But why do some kids get it and others don't? A growing body of research shows that the microbes that live in the human gut and airways shape a child's asthma risk. Could changing the microbiome be the key to preventing asthma? In asthma, a person's airways become inflamed, which causes swelling, mucus, and muscle constriction that make it hard to breathe. Asthma can be life-threatening. In the US, people of color are at the highest risk of developing asthma, and globally, asthma has an outsized effect in low and middle-income countries. Steroid inhalers are the most common treatment, but they don't always work. There is currently no prevention or cure for asthma. IGI researcher Sue Lynch is based out of the University of California, San Francisco. Lynch and other researchers in the field have shown links between the microbiome of infants and childhood asthma.
The human microbiome is the collection of bacteria, fungi, and viruses that call our bodies home. And we have different collections of those microbes at different sites across the body. It may be counterintuitive that the gut microbiome can regulate the airways and an airway disease, but it turns out that microbes make small molecules that enter the circulation and shape the response of the airways.
Lynch and her team have identified specific microbes in the gut and airways that increase the risk of asthma. They are teaming up with a group of IGI researchers based at UC Berkeley, led by Ben Rubin, to create a way to prevent asthma using precision microbiome editing.
We're very interested in how you can edit microbial communities to improve human health outcomes, to decrease negative impacts of our microbiome, maybe even to put in beneficial capacities into that microbiome to improve human health.
Right now, our main tool for changing the microbiome is antibiotics, but they act like a grenade, killing all kinds of friendly microbes alongside harmful ones. Rubin and his team are working to develop tools for precisely tuning the microbiome, targeting specific microbes with surgical precision, something that was technologically impossible until now.
Why can we start to edit microbial communities now? I think there's a few technologies that are really important. One are these CRISPR-Cas technologies, which give us an editing tool. One is metagenomics, which is a way of looking at all the DNA in a microbial community at the same time, which helps us understand what's there and what we should be targeting. And one is artificial intelligence, which I think is very recently changing our editing tools and letting us target microbes and microbial communities in more intelligent ways.
Rubin and Lynch are working together to create ways to precisely edit the genomes of microbes that live in the gut and airways using CRISPR. The goal? To edit a specific gene in a specific bacterial species to stop it from making inflammatory molecules that trigger asthma and other diseases. Human cells won't be edited, just microbes.
Our hope for the next 10 years is that this kind of therapy can be implemented in very early life to prevent disease development in later childhood. This is the right moment for this project because of the work we've done at the Benioff Center for Microbiome Medicine to understand the microbial features that promote asthma, and the work that has been carried out at the IGI developing the tools to precisely engineer microbes in the human system. We believe that this approach is transferable to other diseases and to other body habitats.
In addition to asthma, precision microbiome editing with CRISPR might one day be used to treat or prevent microbial problems like gum disease, metabolic conditions like obesity, and inflammatory conditions like autoimmune disorders or heart disease. These are just a few of the potential applications. As we learn more about how microbes influence human health, IGI researchers will continue to develop the next generation of precision medicine.